Thanks. So, as he mentioned, I'm Brandon Wilbanks. I'm from the Mayo Clinic in Minnesota in the United States and the Department of Chemistry and Biomolecular Biology. And today I'll be talking a bit about some work we've done recently looking for DNA oligonucleotides that modulate biological responses in model neurons. So the disease we're interested in here, primarily just for a brief bit of clinical background, is ALS. So most people are probably familiar with ALS. This is a progressive and always terminal neurodegenerative disease that has a typical course of perhaps 2 to 5 years in people following onset, but some survive much longer than that. This involves the degradation of motor neurons, and then eventually patients with ALS are unable to undergo voluntary motor responses, and this is what's deadly. But what's really complicated about ALS primarily is that 90% of the cases are sporadic. So there's no known genetic link in these people. Somebody just starts presenting with these motor symptoms, and suddenly they're diagnosed with ALS with no background in this. A few or a small percent of the cases have known genetic links. For example, these SOD1 linked diseases have an ASO drug that people are probably familiar with. It was recently approved in the US and in Europe in the last couple of years, but this is only effective in 2% of patients because only 2% have this SOD1 driven disease. And even so, the drug is not terribly effective in these people. It's a marginal benefit. So this raises this kind of interesting question about how do we cure a disease without a target if we don't even know what we're going after. So we work with some neuroscientists and neurologists at the Mayo Clinic who are interested in this. They're Moses Rodriguez and Art Warrington, and Moses in particular has had a long historical interest in ALS and MS. He was actually among some of the first in the world to show that multiple sclerosis has to do with the loss of myelin and from oligodendrocytes. And they took this approach looking for simply just affinity agents that might trigger some kind of response in motor neurons. And they figured that, if we don't have a molecular target, maybe we just search for affinity reagents, screen them for their activity, and ask if any of these have any kind of useful activity. So the way that they did this was they collected IgM or IgG antibodies from patients at Mayo that arrived with monoclonal gammopathy. So these are patients that are overproducing lots of IgMs or IgGs. They purified these from the patients and then simply just expose these IgGs or IgMs to model neurons and ask that they do anything that looks like a neuroregenerative response. And of course, most of them do nothing, or some of them are harmful, but they were able to identify a couple that actually stimulate neurite generation. So here, this is just one example from their paper here. This is one image divided in half here where they printed a fluorescent IgM from one of these patients straight onto a plate surface here, and then nearby we have no IgM plated down. And you can see the neurons here that are growing on top of this IgM are obviously have promoted neural generation, and this sort of starts to look like a neuroregenerative response. So they treated animals with a SOD1 driven model of ALS with this IgM, and they were actually able to find an increased survival, which was impressive, and not many drugs give statistically significant survival even in animals in these models. And they also showed that even though these animals do eventually lose their motor function and have lower activity scores, they can kind of stave this effect off. So this work, working with them, inspired us to ask if we could maybe ask some of the same questions with oligonucleotides, because if we perform selections with really large pools of oligos, we can screen trillions and trillions of solutions instead of just 100 or so solutions from patients. And this work in my lab and Jim Marker's group, along with Nathan's staff, who provided some of the model cells we work with, we perform these selections against model neuroblast cells looking for DNA aptamers that might trigger a neurological response or a neuroregenerative response simply just by their affinity to the cell. Now, these are unmodified DNA oligonucleotides here, which kind of flies in the face of everything we've heard about all morning. So I'll just say here that we think this is probably not a real therapeutic solution in the long term to be all DNA, but it is a good way to start proof of concept because we can do this quickly and cheaply. And if we can identify some targets that we can stimulate with a DNA aptamer this way, we could potentially reselect a stable RNA aptamer later that might have the same effect. So Our process here is a very simple workflow where we just begin by performing 3 selections against a series of counter-selecting targeted cells. So we target negatively selecting against either fibroblasts, HeLa cells, or HEK293 cells. And we have a model neuroblast line where we're trying to select high affinity DNA aptamers against these. So we'll simply sequence them, ask for any that bind to the cell, lower the pool a bit more, and then look for bioactivity like proliferation and neural morphology that's a result of binding events on the cell surface. So we performed 3 selections. I'm just very briefly showing you the selection data here to show that we do see some enrichment. From each of the selections, we have 3 different sets of aptamers we isolated here. So we have around 40 candidates that were selected for their binding to these SHSY5Y cells. And nobody needs to keep up with all these serial numbers. These are just serial numbers that we use. And I'll point out the important numbers as we go along. So here we have roughly 40 DNA aptamers that we're going to screen for activity. And the way that we screened these for binding first was with an IncuSite instrument. So this allows us to place cells down. The instrument will, in an unbiased way, measure fluorescence from the aptamer and will measure confluence on dishes. And this allows us to quantify a per confluence binding activity from the aptamer from images like this, taken across many wells and many plates. And we can use this to rapidly screen all these aptamers. So if we stick fluorescent labels on the aptamer and look for those that bind versus a couple of negative controls in red, these are scrambled versions of some other sequences in here. We do find a subset right off the bat that actually do bind to these SHSY5Y cells statistically significantly. And I don't show the data here, but we also screen these for specificity, and the vast majority interact specifically with the model cells as well. And they don't bind whatever they were negatively selected against. So we take this subpopulation of the aptamers that bind better than both of the negative controls, and then move these kind of down the funnel towards our screens for biological activity. And we're going to look at just a few outcomes in biological activity. We've got colony formation as a very, very easy way, kind of rapid way to screen for any kinds of outcomes. Could be good, could be bad, but we want to lower the pool of molecules that we're looking at. Same for growth rate, and then we'll look at morphological changes like neurite extension and RNA expression changes as a result of treatment and cell culture with these aptamers. So first, we treated these cells for 2 weeks. We place them very, very sparsely. We give daily doses and cell culture media of each aptamer, and then simply count colonies after a couple of weeks. And we have, from this large pool of specifically binding reagents, a few that either decrease or promote colony formation. And at this stage, we are totally agnostic to whether or not any of that is good or bad. We're just trying to continue this screening function. It could be that they slow colony formation because they're killing cells. It could be that they slow colony formation because they're promoting some sort of differentiation response and they're growing more slowly than the initial neuroblasts. And we're just interested in finding those that have any sort of activity. So we quantify these compared to a negative control and then lower our pool a bit more this way. So taking just those that have some effect in colony formation, we also perform just a 4-day treatment course to look for any change in growth as well. So these are cells that are plated at a much higher starting density, and we just asked if they could have any effect over a 4-day treatment course. And only one of these is statistically significant in this much shorter time course of treatment, but you can see the trend roughly follows what we see in the colony formation. So if we could grow these for longer without reaching 100% confluence in the dish, we suspect that some of these effects would become significant. We're also interested in morphological changes that occur as a result of treatment. So again, this is the same treatment regimen. We just give a 250 nanomolar dose of aptamer every day for 4 days to cells in culture. And again, we use this IncuSite instrument, which has a built-in module for neurite identification and measurement, which can quantify either the number of clusters. So these individual cell bodies here are counted as clusters. We can also count the processes. So each individual neurite here is a process, and we can also quantify the number of branches that occur in each case. And we found that one of these aptamers, which we've identified from this screen, actually does stimulate increased branch and neurite length production in these cells over 4 days. I'm going to shift gears to one more aptamer here where we found some interesting changes in RNA gene expression. So these are, again, exact same treatment regimen, once again, 4 days with a small dose of aptamer given every single day in culture media. And then we simply just do bulk RNA sequencing on what's in the well and compare the gene expression we see in our aptamer versus the negative control here. And we get a wide array of genes that are either downregulated or upregulated from the exposure to aptamer. And this is not the case for most of these aptamers or for the negative control. So this is an effect that's really unique to this binding sequence. So if we look among just the genes in the top right quadrant that are upregulated, we then check for Go terms for these. We see a little bit of enrichment for some terms that look like regulation of neural development or positive regulation of neurogenesis. But what we're really most encouraged by is that we see a big boost in oxidative phosphorylation in these cells. And this is considered to be a hallmark of neural maturation. So we see this by both Go analysis of these upregulated genes and by gene set enrichment analysis. We were also, of course, curious what would happen if we move beyond the neuroblasts and move this to some more mature cells. So we worked with Lorenz Studer, who's an expert in the development of model neurons from patient-derived stem cells. And his group specializes in the development of cortical neurons. So they have, over the last couple of years, their group has established this pipeline for exposing different small molecules that they're interested in to developing cortical neurons over the course of a few days, and then screening for biological outcomes that look like neural stimulation or promoting differentiation responses and things of that nature. So I just show here one result from this screen that we found where a very, very small dose of this aptamer that's also causing these Go term enrichment and the neural regeneration and was causing an upregulation in oxalots is also able to trigger a FOS response. So this is a transcription factor that's involved in neuromaturation signaling and neural stimulus. And we don't see the same effect for the negative control. So this is a good hint that we're actually triggering some sort of neural response in these models. We also check for NF-kappa B expression in these cells just to be sure this doesn't trigger any sort of immediate red flag concerning immune responses, and it's not the case that we see this. So at any dose that we can detect the transcription factor activation, we don't see NF-kappa B activation. So we have a handful of things we want to do with our most functional molecules here. Our collaborator Nathan Staff that I mentioned earlier has ALS patient-derived motor neurons and sensory neurons. And that's our immediate next step, is to actually test these on ALS patient-derived cells. We also want to actually perform selections against these neurons. We've done some other work which was recently published selecting against sensory neurons derived from ALS patients, but as these motor and cortical neurons become available, we're going to perform these screens on those as well and ask if we can find oligonucleotides that trigger responses, regenerative responses in patient-derived cells. And kind of in the big picture, another question that we're interested in is whether or not oligonucleotides can serve generally as differentiation reagents, because these things can be expensive in some cases, or there are some cell types that have been historically tough to derive from iPSCs or other types of stem cells. And we wonder if we can just broadly select for affinity reagents against cells that are in different stages of development, if we can find reagents that might drive differentiation in helpful directions. So with that, I just want to thank especially the staff lab who provided a lot of these cells and Mayo Clinic and the DoD